Так, я знову спробувала, зараз подивимося, якщо воно знову зупиниться, спробуємо на Facebook робити трансляцію. Андріан, чуєш мене?
Чую.
Оце останнє посилання, воно вже ніби не вибиває, спробуй його кидай в Slack. Зараз йде трансляція.
Окей.
Я тобі щойно в приват скинула посилання.
Можете додати Марію до паналістів? Бо я не
Так. Я її не бачу, вона додалася.
в attendances.
Зараз.
І зробіть мене ко-хостом, будь ласка.
Та зараз роблю. Я вже тут, дякую.
А тут я не бачу, як запустити рекорд.
Я запустила запис.
Дякую.
Okay, so I guess I should start by now. So thank you for the organizers, and sorry everyone for the little inconvenience we just had. And so I think that everyone's ready. So I should start with the machine learning and deep learning in healthcare lecture. And if you have any trouble, just send a message on Slack or on the Slido. I have it open right next to me. So if you have any problem, just tell me so that we can relaunch if there's a problem with YouTube again. Okay, so to briefly introduce myself, I'm a master's student from engineering school in France, and I have a PhD that I defended 4 or 6 months ago in applied mathematics and machine learning applied to neurodegenerative disease at the Paris Brain Institute. And I'm currently a postdoctoral fellow at the Paris Brain Institute and at the INERIA institution in Paris, and also a deep learning instructor at a company called Le Wagon. And so this is not to speak about me, but just to tell you a few words about my position and how I see machine learning and my scope. So I'm more of a mathematical background with a little bit of computer science. Also, the healthcare domain I've dealt with are more neurodegenerative diseases as Alzheimer's, Huntington, and Parkinson's disease. And finally, my domain of predilection in machine learning are imaging data, deep learning, statistical models, and disease progression, and a lot of mathematical tools. So prior to start, I'd like to say a few disclaimers about the lecture today. It's going to be very broad. It's a very broad and general introduction to different types of data, challenges, and application to healthcare. So as said previously on the course description, I'd like it to be without much math. It's more about the models and architectures that are used in machine learning and deep learning in healthcare, but no deep explanation of how it works and why it works. So it's rather about what we're doing in healthcare, why we're doing so, rather than the how we're using it. The other reason why I'm not going into the details is because for a given task or a given application, there are many different ways to do that. Also, the machine learning healthcare community is separated between two sorts of sub-communities. The first one is the methodological, and the other one is more of the application. So on the one hand, the methodology tries to develop new models and tools to be used, whereas others use them in real-life applications. And as it's more about application, I'm going to focus more on the second part. And as I told you earlier, if you don't know much about machine learning and healthcare, it's going to be a good understanding and introduction to the subjects. On the other side, if you're more advanced and you'd like to tackle such challenges, there's going to be a lot of sources, a lot of articles, a lot of keywords that will help you go through the literature and the different topics there are out there. And finally, I think it's the main disclaimer. I'm definitely not an expert of all the things I'm going to present. I know some of them, and some of them better, some of them less better, but that's how it goes. It's a very broad topic that I try to highlight today. As we're running a bit late, I'll be very brief about that. It's just to be sure that everybody catches what medicine is and how it evolved through times during the past history. So it started like almost 4,000 years ago with different types of traditional medicine. Then medicine itself was more of an empirical study that was trying to cure rather than preventing any disease. Then in Europe, there was some sort of medical winter, as no much of efforts or research have been done in the field of medicine. It was the time of witches and sorcery. whereas on other places of the planet, there was the first medical description and understanding, especially in the Islamic, but also in the Orthodox civilization, with a lot of books about different anatomical structures. Then, in the late 15th to 16th century, the first dissection and anatomy were done in Europe, and first epidemiology models were developed. So that was really back in the past. And what we call now modern medicine only started with the fact with 100 to 200 years ago, especially with hygienicism, that used to understand better and cure better people. We also seen a lot of technical progress that I'll go very briefly about that. The question is, what about now? What state are we in as of today? And this state is very plural. First, there's a lot of different devices as to have medical imaging, but different source of data. We also have a lot of artificial art, prosthesis, and somehow kind of hardware to cure or prevent diseases. There's also a ton of knowledge that has never been so present in the past. And finally, there's a diversity of stakeholders in healthcare that is very huge and large, and that machine learning tries to address. So in a nutshell, you have a lot of information and a lot of progress during the past centuries, but there's a counterpart to all of that. The information got very complex. There's a lot of variability in it. There are different scales of informations, a huge volume of data, and also what we call multimodal information, meaning that you can get the same information from different sources of data through imaging, through genetics, through biomarkers, etc. And this is a huge counterpart, as we have on the one side a lot of applications that we'd like to address. And I'm going to talk about them a bit later on. Aside from the application, there's a huge variety of data that we have to handle and that we cannot handle in the same way. And finally, we have a lot of challenges to use this data. For instance, the anonymization, but also imbalanced data sets, or the problem of false positive, all of these challenges that I'll talk about later also. And so machine learning is somehow at the crossroad of all of that. It tries to address some applications with different types of data by leveraging the challenges it has along the way. And to do that, it basically developed new techniques to handle all of that. So this was a very brief introduction. So the first part of the lecture is going to be about different types of data you might encounter in healthcare and medicine. Also, what are the specific challenges that we have to work with in healthcare, and finally, various applications. And the second lecture will be about more in-depth knowledge about medical imaging data. So most of it will be done without the aspect of medical imaging. So first, let's start with the different data that you may encounter in healthcare if you once will work in this field, which is full of opportunities. The first one that you may think about are basically any type of what we call biomarkers. Those are scalar values, such as a medical checkup, some clinical assessments, cognitive scores, and blood tests. You also have graph, meshes, and network with a different type of data. Then the genomics, also the text, some medical imaging, and time series. So those are the 6 different families of data that we mainly encounter. It's a bit arbitrary to do such distinctions, but those are kind of large families of them. So let's start with the first one, the biomarkers. A biomarker is actually any type of information that you can reduce to one or two number. So a blood test will fill such information, a medical checkup, or any personal information, but also smartphone data that are streamed through those devices. And its representation is basically just a scalar value or a vector, and it's suited for any vanilla machine learning algorithm. So you can use it anywhere you want, basically. A few comments about that. So those data may have two forms. The first one is they may be quantitative. So it's a value that you measure and has some sort of ordering. So the protein concentration, the blood pressure, the heart rate are some of the examples. On the other hand, you may have more qualitative data, such as the gender, the sex, the socio-professional category, some environmental factors, genetic mutation, or a specific treatment. So those are really the basic type of data in machine learning. The other one that you may think of are genetic data. There are two kinds of data, mainly either you take the smallest information possible, which is called the SNP, SNP, or you take a collection of all of the SNPs, and what you'll get is basically a gene. A few comments about them. They are very, very, very high dimensionality. There are a lot of genes and a lot of chromosomes in the human genome. Why they're interesting is basically because as in the past years, they got cheaper and easier to get. That's why more people and communities are working on this type of data. But nonetheless, they're still poorly understood. The problem is not yet to the point of understanding the link between a genotype, the expression of a gene, and a phenotype, the expression of something biological, but rather to understand how the gene itself expresses. It's not clear yet to know why a gene would have an expression or not. And as another disclaimer, genomics, the field of studying genes, are not really considered part of machine learning, or at least not in this presentation. The reason is that genomics has its own convention, their own tools, models, and methods that are quite different from the machine learning community. So here, if we talk about genes, let's talk about them as any cofactors, as any cofactors. So basically as any biomarkers we've seen in the past. In the same way, you can be a male or a female, you can have a gene or not, you can have an allele or not, you can have a SNP or not. But if you're interested, for those of you that are interested about more the field of genomics, there's a very good course on Coursera about those data. Now let's move on to text. So text is, well, it's pretty clear what it is. Some example of text, you can have some doctor prescriptions, you can have the reports of hospitalization or consultation done by a doctor to help other nurses, for instance, to work and to understand what's going on, but also ambulance reports. There are different features you can extract from this text. The first one are basically if you are able to read any values, if the text is structured, if it's a form that someone has filled, then you can directly retrieve some information such as numbers, dates, evolution, whatsoever. The other thing that people start to look at is basically the words that are used in prescriptions or reports. You can lose either at their occurrences, basically those are the frequencies of appearance of each word, or another thing to do is the TF-IDF algorithm that maybe some of you know, and which accounts for the fact that even though some words are very frequent, they are not useful because too frequent. On the other side, some words that are not frequent may be very interesting and they have a lot of information in it. And finally, more sophisticated algorithms, I would say, are the LDA algorithms and word-to-vec algorithms. There are ways or manners to find topics, information, or any sensitive data that you'd like to use in a later algorithm. The main challenges that the healthcare community is kind of facing right now as when using natural language processing and text are challenges that are dealt with not only in the healthcare community. So I'll highlight a few examples of afterwards of the applications that are used for text and some challenges, but mainly the problems that people are facing when dealing with text is a very broad problems dealt with by anyone. Now, there's another type of data, the graphs, meshes, and networks. I'll give you 3 examples of what these are, and I'll let you like 30 seconds to have a guess about what the difference is between those 3. So this is a graph of a brain. This is a mesh of a brain. And this is a network representing the brain. So according to you, what the difference would be between those 3 type of data? So first, a graph just determines the connections between different regions. That's all it is. A mesh, on top of being a connection between different regions, embeds a special location at the node of the graph. And it studies the structure and the deformation of the mesh through time. And finally, a network is some sort of a graph, but where you have dynamic values at the nodes of the graph, and that allows you to find the relation and the correlation and the evolution or different patterns that evolve through time through the graph. And the meaning of those words are a bit arbitrary. They can change from one community to the other. The key aspect is that different communities use different objects that are very, very different, that here would be summed up as graph, mesh, and network. So a graph is basically just a mathematical representation of edges and vertices. That's all it is. A mesh is basically a graph plus 2D or 3D positions at all of the edges. And finally, a network is also a graph, but either the edges or the vertices are valued and potentially dynamically. They can change through time. Most of the time, graphs are basically studied with connectivity matrix. So all of the rows and columns correspond to one of the edges, and the values are the value of the vertices connecting or not the edges. On the other hand, meshes are used more to analyze the deformation, for instance, that will change or transform one data into another. So for instance, here you have the skeleton of an old monkey that is deformed to the form of our current skull. And this deformation, the minimum deformation to go from one to the other, is used to better understand the evolution through time of human. The network is also studied with connectivity matrix, but it has a temporal pattern, I would say. in the sense that it changes through time, and you can have a stack, a collection of connectivity metrics for networks. And for the three of them, so going back to the graph, what people are interested in is basically the nodes of the graph, the connectivity between different positions of the graph, the shortest path between two regions of the graph, nearest neighbors, etc. As for the meshes, the features of interest are more physical features, such as distances between 2 points in the graph, which are real distances in the brain, for instance. But meshes look also at the deformations that can move one graph to the other, one mesh to the other, sorry. And finally, the network, we'll actually talk about that in the next slides. Another type of data is the time series. So some of the time series you might know are EEG for electroencephalogram or electrocardiogram, ECG, but there are also magnetoencephalogram that instead of looking at the electric field at the surface of the brain, looks at the magnetic field at the surface of the brain. In each cases, time series are basically a value that changes over time. So if it's the heartbeat, you have the figure of a heartbeat and the value of the electric response to the heart during the heartbeat. Other type of data include pharmacokinetics and pharmacodynamics. I'm not going to go into details about this type of data. If you're interested, there's a large literature about this data. They're basically tools and methods to understand how a drug or any, mainly drugs, change or affect a person or a chemical substance over time. But basically, time series is any type of the previous data once they are repeated. So if you take a biomarker, then you repeat it, you have a time series. If you take a graph, you repeat it, you have repeated graphs. It's also a time series. And it could be the same for text as well. So if you look at the reports that a doctor would do every month for a particular patient, that would be also a time series. And for that reason, no, not for that reason, but coming back to the networks I've just spoke about in the previous slide, ECG and EEG are actually represented and worked as graphs thanks to the graph theory. They're basically nodes. So nodes are the electrodes, and the values at the nodes are basically the values of the electroencephalogram. And you use this network representation to look at the relation, whether it's a correlation or an anti-correlation between different nodes and thus different electrodes on the brain or on the chest. Those time series are potentially of very high dimension, but of very small dimension. It depends on what we're talking about. They can be of very different scales. For EEG, it's milliseconds, but if you take medical imaging of people during dementia, it can be years. And not a lot of standard machine learning techniques are suited for recurrent data. Recurrent would mean repeated, as in time series. So that ends the part of different data that are used in healthcare, besides the medical imaging data that will be dealt with in the second part of the lecture. So now that we know what data we're going to work with, let's look at what are the challenges that are specific to healthcare applications. There are a lot of challenges that are true for any type of machine learning applications, but those are specifically the one you will deal with if you ever work with this type of data. Okay, so that's basically what I just said. And on top of that, there are some challenges that are particularly specific, not to only healthcare, but a given type of data. And finally, there are other challenges that concern more legal aspect that I'd like to talk about. So let's see them. So the first thing that happens in a lot of data sets is the high class imbalance between the data you have. And it's a real problem. So let me explain with an example. Let's say that you work on Alzheimer's disease, which is supposed to be a very common disease. But there are only 30 million people out there out of 7 billion that have Alzheimer's disease. So it makes it 3 out of 700. So if you have a classifier that works on any data, any patient around anyone in the street, with a classifier that tells you no Alzheimer, you'll have an accuracy of 99.6%, which is very high. So what you have to do with your new algorithm is to be between this value and 100%. And this makes the comparison of different algorithms very difficult because they very depend on the number of patients you have within each class. The second related challenge is that you have this high class imbalance, but on top of that, you have very small data sets. As I told you, Alzheimer's disease is a very well-known disease and very widespread, but there are less prevalent diseases such as Huntington's disease, where you have only 1% out of 10,000 in Europe, for instance, and it's very rare to collect enough patients to include them in research or cohort studies. And even if you do so, let's say that you have a first research cohort, then a second one, then a third one, etc. You really believe that the same individual would like to be part of several cohorts through time and go back to the institution and go back through the test and testing some drugs. So it's very hard to try and to work with this type of data. Also, we have to compare the small data sets to the very high dimensional features that we're working with. So let's say you have a cohort of 10,000 patients. That is actually already very, very impressive. Aside from medical imaging data, in general, having 10,000 patients is quite incredible. But now compare the 10,000 patients to one single MRI, which has 16 million voxels. So 16 million information within one image of one patient. And it goes the same for the genome that has 3 billion pairs or 22,000 genes. Same goes also for the EEG that have 60 electrodes that records data every millisecond and has different frequency bands, and that goes from 250 to 2000 Hz. And finally, if you work on the neurons, the brain has between 30 and 100 billion neurons for one single individual. So you always face the fact that even if you have a lot of data and a lot of patients, which is already amazing, you'll have to face the fact that any sample of information is larger than that. And it's going to be hard for you to know whether the information you have is particular to the subject or not. The other challenge that might occur is the false negative, false positive problem. So it is true for any application in machine learning. You never want to have a false negative nor a false positive. But if you think about real-life application, saying so to a patient is very, very different in the consequences it would have. What if you tell someone that he or she has a cancer while he or she has not, and vice versa? The implication can be very disastrous in both cases, and actually depends on the application. Sometimes it's better to tell something rather than not, but it can be also the opposite. If you start a chemo or immunotherapy, whereas you don't have anything, it can be damages for your own body. This is a point I quite like, because it's something we don't think about much, but that we deal with all the time. It's probabilities. Our brain and our thoughts and what we think about what's our environment and what's around us is not suited to understand probabilities. So what if I tell someone that you'll have 67% chance of having a heart attack next year? What does it mean? Will you have it or not? And what if it's 73 within 2 years? It's plus 7%. What's plus 7%? So it's very hard, and it's something that we need to work on as patients or practitioners, as it's not something we have we deal with usually. You basically have a disease or not. And it actually goes the same for almost everything in life. Another challenge, as I'm just going through all of them, is the fact that sometimes you don't have any ground truth. So let's take, my favorite disease, Alzheimer's disease. The only fact that we can't prevent it comes from the fact that we don't know it much. It's very poorly understood. And the definition of Alzheimer's disease is changing over time. It's getting more and more accurate. And so the doctors around the world or around institutions have different definitions that they sometimes and often actually disagree on. It's not that they disagree, but they just don't have the same experience and the same meaning under the same word. So the labels that they give to the patients, and so the way the data are labeled may be very different from one practitioner to the other, making the label not 100% correct, or not correct at all if we don't know the ground truth. And finally, this is a real challenge that is very interesting because it's more of a machine learning problem and about the community. There are not many persons and labs around the world that started to work on multimodal data. And by multimodal data, I mean using different sources of information for the same individual. So for instance, using the MRI data, but the genetics as well, some biomarkers, some cofactors, some time series, etc., to have a coherent representation of the whole patient thanks to different data. And this might help somehow dropping or not taking into account the noise that can be particular to given data, but not the other. The more information you have, usually the better. And finally, just a quick word about missing values. So missing values is a common problem in machine learning. And I'm not saying it's even more common for healthcare, but the solely fact that a value is missing in healthcare might be some sort of information. Let's say that you have a huge data set with all of the tests that a population might go through. The only fact that a person hasn't been examined for a given disease might mean that he or she doesn't have the disease because the practitioner doesn't tell the person to go diagnose himself or herself. And so this literature is about what we call being missing at random. This is usually what occurs. But in healthcare, usually data are not missing at random. The value of the missing data is actually related to the reason it's missing. So if you don't have, for instance, a diabetes test, it's probably because no one thought it's relevant for you to go through this test. Now, another one. Let's say that you are working in a temporal setting and you're looking at someone evolving and progressing through time. Now, let's imagine that you will give to this person a drug in a clinical test to test whether the drug is efficient or not. You can't tell, at least from just the observation, whether the progression of the disease is accelerated or made slower thanks to the drug, or just it has no effect. Because to know that, you'd like to compare what would happen without the drug and what happens with the drug. And it's not something feasible in the case of, well, clearly, as we're all different, it's not feasible. That's basically why, this is one of the reasons why there are in clinical tests a placebo arm versus a treated arm. Now, a question that you should ask yourself when you work with machine learning problem is, who is this diagnosis or prediction made for? You have a final value, you're able to predict any dementia, any cancer, any disease, any patients stay at the hospital, whatever. Who is this beneficial for? But more importantly, who might suffer from such information? Do you really believe that saying to someone that he or she is going to die within 10 years, 7 months, 3 days, and 2 hours is relevant? I mean, those are more of ethics questions that have not been looked at very carefully by practitioners. There is a community that works at this type of question, but there's no much link in talks between both like practitioners and those that are thinking about that. And this last challenge is related somehow to the first. You can't compare one individual against the same individual with or without a drug. The same question applies to comparing two patients. Let's say that you have some data, the size of a bone or of the brain or any size. It's actually size are good examples. Let's say that you have two persons in front of you, one with a big brain size and the other one with a smaller brain size. Do you know whether the reason why the small brain is smaller? Is it due first because some people have a smaller brain, or because it's the consequence of a disease? And such information and the fact that you have data, raw data, doesn't mean that you can always compare them straightforwardly. And this is a very important question when you deal with healthcare, to know whether someone is ill or not. Okay, another one that I really like is usually when you read about AI almost anywhere, you get this headline, AI is better than doctors, or AI is actually better than anyone. A small parenthesis, AI is better for a lot of things, like my computer is able to remember a lot of movies, like terabytes of movies by heart, second by second, or actually 24 seconds, 24 images per second, whereas I'm not. So computers are already better than humans for a lot of tasks. But AI is better than doctors. So you should be very, very, very careful when you have this information in front of you for two reasons. The first one is the tasks that are compared the doctors and AI on are tasks that doctors are not trained on. Go to your favorite neurologist or doctor and ask him or her to predict what's going to happen for you in 10 years, even in 1 year or 2 years. He or she is not able to do that at all because he or she is not trained to do that. And it goes the same for a radiologist. They will say, we'll look at an image, and if you ask him, hey, can you tell me where the blood vessels are? And like in 1 second and all of them, this is actually hard to do. And they're not trained to do that. And so this is one of the main, not the main, but it often appears that doctors were not trained to do a given task when they were beaten by AI computers. The other reason is, and it strikes me a lot, is that how can you be better than doctors as those tasks are measured on ground truth, and those ground truth have been already labeled by doctors themselves, not the one that done the task, but other one. I mean, the ground truth of most of these challenges is a ground truth that has been labeled or diagnosed or predicted by the doctor themselves. So AI is better than some doctors, but you can't beat what is supposed to be the ground truth. This is another challenge I'll speak about in the second lecture, is the problem of going from research to clinics and even to real life. So many scientists are interested by academic research, but there is no incentive to show that what you've done as a proof of concept works on other data sets, on other cohorts, or in real life data. But I'll go back to that later on. And I'll skip, okay. I'll go a bit quicker. So some of the challenges that are specific to some data. So for EEG, so remember the time series that are recorded on your brain or on your chest for the cardiogram. They are very high dimensional, as we said earlier, but there's also a lot of noise. Usually when you have a sensor that gets some signal through time, it's fixed, it can accurately reproduce the signal. Whereas here, in this setting, the electrons are very hard to position. Their position can change from one person to the other. There can be errors, and there might be moves from the patients during the session. And finally, this is a very interesting one in the scientific community, is once you have your signal, and you're asking your patient a specific task, so for instance, to raise a hand, or move an arm, or move the leg, or speak, or think, or learn, and you have your EEG, at the end of the day, how can you discriminate between, in the signal, what's related to the normal and stochastic state of the brain, called the resting state, to the specific task at hand? You could do that if you have not a lot of data, a lot of patients that do the same task. But once again, the EEG is very high dimensional and you don't have a lot of data usually. There are a lot of patients usually. Other challenges specific to time series, but not EEG time series, but more longer time series. Those are cohorts that we call longitudinal. It means data that we see a repeated number of times. So a patient that came to the clinics or the hospital 1, 2, 3, 4, 5, 6 times. The problem with such data is it's very hard to maintain those cohorts during weeks, months, even years. And you have to maintain the same protocol from practitioner to practitioner to be sure that once you read a data, any biomarker, for instance, a cognitive assessments or on a given person, it has been measured in the exact same way for 2 different persons. This is also a bias that people don't talk about much, is the fact that in those cohorts, when someone's missing, it's again not at random. if someone misses a value, there's a reason to that. Maybe he or she is not able to move and to come to the clinic because of the disease. Maybe he or she is not just dead. And those are reasons why there are drops in the data sets that are absolutely not random. And a final issue is that for some diseases, such as neurodegenerative disease, they last for so many times, they appear for a period of times over 5 to 10 years, that makes it difficult to follow within one cohort. You have to keep the cohort running for 10, 15, or 20 years. It's a huge amount of money. It's a lot of infrastructure. It's usually multicentric, meaning that you'll recruit your subjects in different places. Now, as for text data, you probably know that doctors are not the best writers. So there has not been yet, to my knowledge, handwriting recognition done well for doctors' handwriting data. But also, when the doctor is writing, or anyone is writing a prescription or a report, you have to know that it's written in a specific context, for a specific reason. And one of the worst reasons is that in a lot of countries, you get refunded or reimbursed based on what you've written, based on the prescription. So it's really common for a doctor to add words that are not 100% related to the data you have or to the observation he or she will have. And finally, once he or she is writing what he or she is having in front of him, is not everything. When you're a doctor, you're not a data manager or data collector. You have someone in front of you and you have to deal with the situation, with the person, and you have no reason to write everything down, which makes data incomplete and not prone to give you all the information you have. And usually text data are the data that are the most commonly used in accordance or in relation with other type of data. Oh yeah, I'll just skip this one, or we're running a bit late. And I'll go to different applications in the healthcare. I'll go through different applications done by, thanks to machine learning in healthcare, except for medical imaging that again we'll look at in the second lecture. So first, medicine did not wait for machine learning to start using math. There's a hype in the journals or everywhere about machine learning in healthcare, but healthcare has long ago started to use math, first with different types of biostatistics, in particular epidemiology models that you may have heard of during the COVID pandemic are not machine learning algorithms at all. It goes the same for survival models that you can look at if you're interested, but also genomics that I talked about earlier. So as for the applications we look at, there are so many of them and so many different. I first tried to look at the questions you asked on the Slack channel so that I've included some of them. And I've tried to distinguish some of the families, but it's very arbitrary and you can take, well, you'll just look at them. The first category in the first family are the ones that are related to clinical trials and drug developments. The second one are those related to natural language processing. Then those that are related to EEG and ECG. Then obviously you have all the applications with medical imaging that we'll talk about in the second lecture. And finally, I'll give you a focus on one model of disease progression. Yeah. So first things first, drug development and clinical trials. Sorry, what does it mean? So healthcare is more than just doctors having patients. It's also developing new drugs for diseases to cure or prevent given diseases, but it also not only developing the drug, but developing the environment to prove that the drug is working. This is basically the clinical trial. You are trying to prove that the drug is working in a given setting and environment. So first, there's interesting literature about how to propose new molecules designed with ML tools. So either you write constraints on the molecules and how they're built, and you let an algorithm run, for example, a Bayesian optimization that will try to have new molecules that you haven't seen, but that comply to the rules that you've intended and given to the model. Or more recent techniques that have been using GANs. So those are generative adversarial neural networks that generate data that somehow look real. They generate data that are designed by design to look real. And so this is another way to create new molecules that might be real without predefined rules, contrary to Bayesian optimization. Another application, okay. Okay, so another application is once you have a lot of different molecules that have been released and a lot of different diseases that you work on, it happens sometimes that a molecule that you targeted on a particular disease actually works on another one. And this is something you did not predict. But this is something you can look at thanks to the large literature, scientific literature, and you can mine all the texts that are in the literature and see whether there are relations between diseases and molecules in the literature. That happens a lot through probably another disease or another gene that co-occurs, and maybe try not to develop the drug, but to test the drug on a disease that you haven't thought of. Another application of in the case of clinical trials is the cost effectiveness of those trials. Usually what happens is that you recruit a high number of subjects, you scan all of them, and you subselect only the one that you're interested in. So in the case I'm just presenting, you're interested in subjects that have a particular feature on the scan, but you have to go through the scan to know whether he or she has the feature. Well, the way to do that is to predict based on other less costly values whether the person will have or not this particular feature. So for instance, you have persons with dementia, again, and you'll try to predict whether he or she is demented prior to make a scan of this person and then select him. And in that sense, you will lower the number of scanned subjects and thus the cost associated to scanning them. And basically, you do not only reduce the cost, but also the burden. Those medical imaging machines are costly, but they are also under the pressure that there are always persons and patients trying to go within the machine, and it's very scheduled. And so this could be a reason to release the pressure on those machines. And finally, a last application in clinical trials is once you have the response, as I told you earlier, one of the challenge in when you try a new drug is to know whether the drug you are giving to the person has an effect or not. And what can happen is that it has an effect that is differentiated between different persons. So instead of having one effect for everyone, it has different effects for different persons. And in this context, there have been quite different approaches and techniques trying to leverage the fact that the treatment can have different effect on different subpopulations. And you want to look at those different effects and dissociate them. And then you can also look at what are the cofactors, maybe genetic cofactors, that are related to the fact that you belong to one treated effect or the other. So this ends the application for drug development and clinical trials. Now let's move on to different applications in the field of natural language processing. A known one is basically to have an NLP chatbot to guide a patient or the doctor or the staff to direct the person to a given department, for instance. And this can be used to prevent a person from going to one person to the other, from one administration to the other, and going to the right person at the right time. And the right time is usually now, not later. However, it faces some problems, as those type of applications are usually, especially for the wrong prediction, very wrong. So usually there's still, after the ML procedure, an expert system that says, if you have that, you can go there. It doesn't make any sense. Whereas the ML algorithm can have a very misleading prediction. So all the systems that work in practice that I've heard of have always a second layer back off with an expert system. Another NLP application that is actually working in real life in some parts of Denmark, and hello to the one that are from Danish here. So an operator, when he or she's called for an emergency, he doesn't know what is happening. And it can be very hard to get the information from the person or from the person that is calling for a variety of reasons. And so there are NLP bots that are listening to so they're listening to first what the person is saying to the phone, but also how they're saying it, their pace, their breath, etc., to better know what is possible that might have occurred and what are the best questions to ask to better know what the real problem is. So it's kind of a decision support system that operators can use in the context of emergency to precisely know what is happening. A third application of NLP is the one I've discussed earlier. I don't know why it's twice, but it's basically the relation between. So I've discussed the fact that you can try a drug on an apparented disease or a disease that appears a lot in the literature and that is related to it by some scientific papers. You can also do the same, not for a drug, but really to tackle the link between a gene and a disease. If the same relation appears a million times in a million paper, there is likely that the link really exists. And those paper usually starts with the word somehow meta-analysis, meaning that they haven't done any study, but they've analyzed the paper that all say do the same analysis. And this helps also to find the false positive from the false negative. It happens that some relations have been found, but only in one paper out of 10,000, while if it's only once, it's maybe a false positive. And a last application I've heard of probably a month ago is that if you listen to how a person speaks, you can detect whether he has a particular disease, in particular dementia, that prevents you from speaking properly. There are some dementia that affects some part of your brain with which you cannot speak as before. You'll use other part of your brain to speak, which makes your speeches a bit different. You'll use other words, other metaphors, other semantic fields to speak. And from that, there are actually applications that tries to detect whether you have or not a specific dementia. So from these features, it can retrieve the type of words you're using, their frequencies, the grammar you use, the syntax, and finally, what it does just afterwards is to cluster or to classify those features to know which group and which class you belong to. Someone asked me to speak about, also the application of time series as EEG and ECG. To be honest, they are very, very similar. I mean, they're all trying to do the same, is to basically understand how a brain structure or function has been changed or damaged under a given condition. And it's trying to do that based on the connectivity between different regions. So you probably remember that I've made a clear separation between a graph and a network, which might not seem always relevant. But this is a scenario where it's important to know why such a difference exists. A graph is basically know what's the topology, not in a mathematical sense, but more in a physical, like what is the topology and the structure of the brain? What are the regions that are connected one to the other? And once you know it, you can use the structure to look if the functions that go through this structure are the same for different patients. So if you know that on average, two regions are highly connected, maybe sometimes they're not anymore for particular diseases. And this is done thanks to networks that are built on graphs. So this is one of the interesting papers that have been written 3 years ago about the loss of connections and of functional connection between two hubs of the brain. A hub is an edge that is highly connected in a graph. And so such connection has been lost for patients with Alzheimer's disease. And another example is for, I don't even remember. Yeah, okay, it's for drug addiction, yeah. So getting some drugs, here it's our opioids, may change the connections that are in the brain. And in that spirit, I haven't found the paper, but some psychedelic drugs are actually increasing the relations between different parts of the brain. And this is pretty much why people have hallucinations or strange feelings, is because they're different parts of their brain that are not used to be mixed up together and have similar pattern at the same time. Well, under drugs, it can happen that two different areas are simulated at the same time together, which makes this feeling. Now, this is also a very vast field called brain-computer interface. It's basically how, thanks to electroencephalogram and thanks to different algorithm, one can control a computer, which is very interesting. And so there's a large field about that. I would say that there's a real difference between what is proven in practice in science and what's written in the media. The experiments that we are actually working on are basically a Pong game where you can get up or down and try to stop the ball when you're playing against another Pong player. And that's the most basically we can do for sure with 100% accuracy. Whereas the other topics are not yet solved. And the reason is not that we don't know how to do it, it's just that the brain is so complex that we don't know which information to retrieve. So as for now, the science is more at the pace that not at the pace, but as for now, science is just trying to understand when you do a given task, which part of your brain is activated and how it relates to the other parts. And finally, this is a funny one that they really much use is when you put chest electrode for an electrocardiogram. There are between 4 and 8 of them, I guess, depending on the country and the system. Or also when you put an electroencephalogram, when you have 64 to 72 electrodes, I think, there might be some confusion and you might invert some of them. And this is a real problem if you are under a condition or a disease and you have to know right now what the profile of the signal is. And so there are algorithms that, based on the proximity of the signals, can tell you whether the two electrodes has been inverted. Another type of application that I didn't mention is the precision medicine, which is, to me, more of a buzzword as for now than something real. There are not a lot of applications currently. There are a lot of under investigation, a lot of research. So I hope that within the next 5 to 10 years, it will get true, but now there are more of proof of concepts. One that is very interested is appears in the case of diabetes, where there's a almost known relation between different sensor factors, such as the weight, the blood rate, heartbeat, etc., to a potential vascular problem. And so sensors are used by the patients, but they are controlled and monitored by a doctor that can right away call the patients to tell him or her to come to the clinic in case there might be a potential problem under the next 24 hours. And the second example that is also now widely used, and it was actually one of the project of the data science summer school 2 years ago, made by the Lviv hospitals, is to basically predict the length of the stay and the survival time and risk within a hospital based on the emergency and different factors and reports. And it's useful for a lot of different reasons, depending on the system, depending on if it's used by the emergency or the hospital or the patients, etc. So there's a bit of literature under if you want to look at it. Oh yeah, and this is some data I've been working on recently. This is in hospitals in France. There is a very high seasonality at the emergency entrance between Tuesday, which is a working day, where there's a lot of persons coming in, and where there's almost no one on Saturdays and Sundays. And the ratio, I believe, is 1 to something like 8 or 10, which makes actually the work at the emergency, like the management of people at the emergency, very complex. And finally, there's a last one I'd like to mention. It's not a real application, but it's all the challenges that are out there. And for those of you that are less skilled than the other that want to learn, there are many challenges on Kaggle, but also anywhere else to learn with various type of data, so you can force yourself to work on EEG data or MRI data or biomarkers or genetics or anything you want, based on the And so this is one of the example. It's called tadpole. It's prediction of Alzheimer's disease. It's prediction of the label, whether the person will be cognitively normal, CN, MCI, which is the first step of Alzheimer's disease, mild cognitively impaired, or AD, Alzheimer's disease. And there were other prediction, such as cognitive scores. So what is your cognitive decline within 2, 3, or 4 years? And what are the volumes of your ventricles with the brain structure over time? And so those are all the participants. And actually, this is very interesting because as the challenge worked very well, we actually won one of the first prizes within the team. We were invited to publish a paper and also to share our algorithms. And now there are 10 or 15 algorithms that are shared so that anyone can use them on real data. And so again, it's really a real opportunity to master machine learning, deep learning. You'll learn mostly once you've go through all the different concepts and theory, you'll master it only by practicing and practicing. I'll take last minutes as we started a bit later to speak about one type of model called, this is a family of models called disease progression models. And to illustrate why we're using that, I'll start with such weird illustration and graph. When you have an image, we'll talk about that in the next lecture, but think about it. If you have an image of your child at 6 months, and you have the same image of the same child at 12 months, if you average the image, as if it was the picture at 9 months, the Euclidean mean, the mean of all the pixels, would result in something strange, right? So you cannot average images one with the other. However, you know that within the space of all the images of this size, there is the image of your child at 9 months, which is here, this point here, which is maybe not far away. Actually, it's a very high dimensional space, so it's very far away, but it is somewhere in the space. And so this curve here, blue and red, is basically what we call the space of possible observation. Out of all the possible images, but also you can think about any value you want, not all of them are possible. There is a subspace, which is the space of all the possible measures. And on this space, it's somehow possible to speak back about mean, about operation, about subtraction, about direction, which was not possible in the Euclidean space. So here what happens is that if you have not only one baby or only one patient, but many of them, this is what you'll get. So this is a first gray person that came 3 times at 3 different visits. So in blue, it's a patient that came 5 times at 5 different visits. And green, a patient that came 4 times at 4 different visits, right? And so what it tells you is that there's a space that shows how the people are evolving and from which we can somehow predict what's going to happen next. And on top of that, there's what we call the average trajectory, which is basically the average evolution once you've erased the variability within the people. As some persons might start their disease earlier, some later, some faster, some slower, some with a different pace. There are a lot of differences between individuals, but once you've withdraw it, you are left with some sort of an average trajectory of the disease. And so the intuition to build such a model is basically that you have your person that came 3 times, you have 2 other patients, Yij is the observation of patient I times Tij. So again, 3 times Ti1, Ti2, and Ti3. And what we do is we're able to recombine all of the observation within one long-term history of the disease. This is the disease progression model at the average disease progression. So once we've done that, we're able to go back from the average trajectory to the individual one. And the reason we're doing that, thanks to the individual parameters, is basically to compare the group average trajectory to the individual one, whether the person goes faster or slower. It has a lot of different consequences. For a doctor, for instance, you may know whether to see your patient within 1 day, 2 days, 3 months, 4 years, depending on the pace. So those parameters, that's some of them for some models, but deal with the fact that some start earlier, later, slower, faster, and also that the, how to say that? The sequence of events that appears during a progression is different from one individual to the other. That changes the ordering and might differ from one to the other. Let's skip all that part, sorry. And finally, as we're able to do that, we're also able to drive the future of the mean to know the future of the individual. And that helps us to predict what the disease progression would be, not at the population level, but at the individual level. And so the objectives of such models that are worked on are threefold. The first one is to better understand for a doctor or practitioners the disease progression at domain level. The second one is to monitor individual trajectories and to compare them to the mean. And the third one is to have individual prediction. And this is very important for clinical trials. As I told you at the beginning of the lecture, you don't know what happens, whether you don't know when something happens, whether it's due to the drug or the disease. Whereas if you have a given prediction, you're able to know what's going to happen without the drug and compare it to the case when the person has the drug, and to see the effect of the drug at the individual level rather than between the placebo group and the treated group. And so the advantages of such model are that you can use very different types of data within the same framework that helps you describe the data you're looking at, but also to personalize the model to the individual and to study which are the subpopulations that go faster or slower, and to analyze the impact of some genetic factor or environmental factor or socio-demographic factors on the pace of progression, the age at disease onset, years when the event occur, etc. And finally, you're able to predict what is happening for the person. And so to conclude, I'll just show you what such model can give. So here, what we are saying are the four different modalities. This is the cortex, the size of the cortex, the thickness of the brain, of the matter that is around your brain, that changes through time, that gets smaller and smaller due to the Alzheimer's disease. This is the mesh of the hippocampus, which is basically part of the memory. Here on the top right corner, you have the PET data, which is the consumption of the glucose through time. And once the brain is dying, it's using less glucose at different regions. And here are the conversion from cognitive scores from normal to abnormal value. You have a normal memory that converts to an abnormal memory value. And once you have this disease progression model that can be done on different types of data, you can personalize it to one individual. So let's say your practitioner, you have new data, you can feed this model to this data by saying, okay, this person goes way slower or faster, or it starts the disease 3 years earlier or 3 years later, or maybe that the pattern or he or she, the event occurs in a different order for a particular person. Because again, everyone is different. So this is the type of model that are actively used or developed in the community. And I think, yeah, that ends the first part of the lecture. So I think that's the point where when I answered the questions, the first one would be, could you please make the slides of the lecture available? It would be really nice to have them as reference for future studying. Yes, of course. No problem. Yeah, you recommended the course on genome analysis at Coursera. There's a huge range of courses. Which one would you recommend? I think it's the one from the Johns Hopkins University. And who is the other? Well, the Johns Hopkins University, I think there are many speakers. What about epigenetics? Their study to link change in epigenetics to change of different biomarkers and therefore way of life. I'm absolutely not an expert of epigenetics, as it does not involve much of machine learning to my knowledge. So I hope it will, but I have no idea what's the current impact of epigenetics on the phenotype of the individuals. Does GPT-3 get a use in analyzing of medical text? I don't know. So again, I don't remember if I said the question. Does GPT-3 get to use in analyzing a medical text? From the articles that I've read, using NLP in healthcare in medicine, barely half of them are used with neural networks. Most of them are using word frequencies or other handcrafted features because they rely on the fact that they have to interpret the data, for instance, in the disease gene correlation, which exists in text mining in the scientific literature. However, all the startups that are working in the medical field, especially those that are analyzing the voices of the patients or their records or whatever, there's a first part which is a speech to text transposition, and a second part that is a NLP, and I would bet that they're using the last NLP models, but I don't know. I don't know if it's a good enough answer. Okay, so there's an answer, a question from Yaroslav Kuzemko. I was told that genetics doesn't use machine learning tools. The previously Stanford course for ML that told that unsupervised ML has been used in genomic analysis. Here's a link to the screenshots. So yeah, this is one of the very, very few examples of ML in genomics. And I think, I'm not 100% sure, I've actually removed this feature from my presentation. I think that is that one. It's one of the first examples of ML being at the front page of the Nature journal. So basically, depending on the community you're speaking to, you can or cannot say that genetics are part of machine learning. They can be, as the methodology and part of the algorithms they're using are very similar, but the way they're using them is a bit different from standard machine learning approaches. And so for that reason, well, there is a separation between them. So for instance, if you look at the, okay, one of the, I'll give you one example to make things clear. One of the very well-known algorithm used for genomics, or at least genetics and relation between a gene and a phenotype, is the GWAS. And if you look at what the GWAS is, it's basically just a bit more complex linear regression that takes care about all the covariates. Do you consider it as part of ML or not? I would say yes, as it's the same tools. I would say no, it's the way to use it is quite different and is used only in genetics. If you believe that ML is a tool rather than an application, then the tools used by genetics are not the tools that you can use for other domains. If you do not agree with my question, do not hesitate to PM me or ask another question. I would be happy to discuss it, or even on the Slack, actually, that is somewhere around. Could you please, okay. What is your opinion and last progress in use EEG for early diagnosis of neurodegenerative dementia? Well, there are a lot of literature about EEG for early diagnosis of neurodegenerative diseases. The problem that there's a fundamental problem that I'll address in the second lecture is proving something on some data doesn't mean neither that it's valid, then you can replicate it, and then you can transfer it to real life data. And so there's a lot of papers that have proven that you can use EEG for those diagnoses, but it has been done on 10, max 15 persons, sometimes more, but not a lot of studies, and it has not been replicated. But this problem is inherent to a lot of research applications, not only EEG. Could you please recommend a good intro into time series analysis for neural data? It depends on, okay, so PM me or send a question on Slack about what time series you're talking about, either if it's EEG or longitudinal data or anything, because those are very, very different topic handled by different models. Any comments on Elon Musk Neuralink? The only fact that there is no more comments about Neuralink for the last 2 years or a year would be my comment. Seriously, I mean, I wish him good luck, because there's a lot of interesting research applications along the way if they're seriously going to do what they're saying they're doing, apart from the real application and motivation. Nonetheless, they are only in Paris, there are 4 to 5, even 6 companies that are working at only understanding the brain and how to translate the signal of the brain into something meaningful. So only that part is very complex, and people have been dealing with it for 5 up to 10 years. And so Neuralink is promising something that is way bigger than that. So I don't see it coming for the next 10 years, but we'll see, and I would be interested. Okay, I'll share the Coursera course. Okay, I'll be honest, I've never heard of folding at home, but I'll get a look at what it is. Do you know if Big Pharma make use of this project for drug discovery? Okay, I've started recently working with pharma companies, and from what I've seen is that they may be part of the companies that are thinking about machine learning projects, but from the one that are proving data and doing clinical trials, they're still at the biostatistic, more not ancestral but classic methods. The reason is not that they don't know a more recent method. It's more than the regulation agency do not allow yet other type of proofs for drugs. I mean, as for now, having, for instance, for a drug clinical trial, having a treated arm and a drug arm is still the holy grail. What about Apple Watch, ECG? Can the wearable device be the next step in healthcare? A lot of companies are, okay. So the question is about wearable devices in healthcare. A lot of companies are thinking so. So the fact, there's one thing that they didn't thought of, and a challenge they are facing that is not related to healthcare is to the devices and sensors. Being able to get a signal from a sensor or the device is not as easy as it seems. And so those companies are now just trying to use those informations. And moreover, there are many questions about such devices, such as how could you prove that a device is better than a gold standard, as you can not beat a gold standard in proving something. So before to use it in real life in a wearable, you have to prove it on someone. I mean, in a research environment. Based on your experience, do classical ML methods ML models work on medical data, or usually neural network work better? Okay, this is an interesting question. Not interesting only for medical data, but in general. This is an advice I give when I teach deep learning. If your data doesn't require you to use a neural net, don't use a neural net. If you have a matrix X and a vector Y, and you can use a SVM, a random forest, a Bayesian model, whatever, use it. Why? Because scikit-learn allows you to test 10 models, 10 different models with millions of hyperparameters just in a click. And on the other hand, you'll try a neural net, you don't know how to design the architecture, you don't know how to design the layers, the neurons, the activation function, nothing. So it will cost you a lot of time, maybe for nothing. So if you have data that you can work with easily, go for an ML model. On the other hand, there are data that have been tried to be handled by ML models that cannot and are way easier to handle with neural nets. Those are medical imaging and imaging in general, time series, and so recurrent data, and with the surround and text. So for those three, it's very hard to find a standard ML model that is suited for those type of data. So in that case, don't use scikit-learn because actually you can't, and use a neural net. Yeah, setting a Keras neural net for image is like 10 minutes. Whereas if you want to use an image in scikit-learn, good luck. What is your opinion about using machine learning for a diagnosis such disease which do not have any physical or structural changes, such as autism, autistic spectrum disorder, schizophrenia, etc.? Which type of data and tools can be used for this purpose? So actually, I don't really know what you mean by no physical or structural changes, but there are no phenotypic changes, but there are changes seen visible in the EEG. So there's also, if you want to work with autistic spectrum disorders and schizophrenia, I would advise you to try first EEG, as they provide data. And I know that there have been papers about such data, such discovery for this type of disease. There are also other diseases trying to look at whether there is a structural difference within the brain in schizophrenia, but, well, I don't know what the definitive answer is. Is there any news on the folding? Okay. Again, I don't know what this is, but I'm going to check. No problem if you couldn't watch the whole lecture, it will be available as the other one, no problem. What about developing an application using natural language and computer vision to help the guys at the medical store automate the process of selling medicine? I'm not entirely sure what you're referring to. Do you mean that you go with your prescription, you show it to a computer, and you have your medicine? Well, you can imagine that. Yeah, I've actually skipped the legal part of the challenges that you can. I'll give you the lectures so you'll be able to look at it again. But structurally, changing something in healthcare is very complex. So if you want to go to the drug selling store and automate it, it would be complicated, not for ML purpose, but for regulatory purposes. But I'm not sure I perfectly understood the question. Is it your second time at this school, according to the signing under the photo on the first slide? It's actually the third time. First time in 2017. Maybe this is not very related, but maybe you're faced with symptoms recognition on video. Okay, I think this is related to another question. Yeah, okay. So there are algorithms that are that looks at your expression and tell which mood you are in. And you seem that it's not very relevant, but actually it is for some dimensions where you don't express some expressions like anger or happiness or joy. And there are also recommending system in airports to code temperature or flu of the participants. But I don't know how they're working actually. Is there any papers on applying Bayesian approach to noisy sensors in biomarker analysis? Will it prove better than frequentist approach in your opinion? Okay, so Bayesian against frequentist, first in general, then in noisy biomarkers. I would say that somehow healthcare and medicine, due to the fact that the information may come from different various fields, is the dreamed field of Bayesian statistics rather than frequentist. First, because you don't have a lot of data, you have a lot of a priori from knowledge, from expert knowledge that you have around you, but also because often with Bayesian approaches, you can have generative models that you can't often have with frequentist models. And in that sense, it's people are interested in the Bayesian approach. But I'll talk a bit about that in the second lecture as there is a problem of vision and of understanding of what the models are and what the results are and what should be said from our perspective as scientists and mathematicians and computer scientists and practitioners and computer practitioners. And our point of view is very different from the one from doctors. And for that reason, there's a misunderstanding between what they and us are doing. But I'll talk about that. Hi, can you please comment on data augmentation for time series datasets? Usually classes are imbalanced, but is it correct to improve it with augmentation? For example, we can split long ECG signal of one patient to more short periods. So, okay, again, voice, it rings a bell for different things. So for ECG, electrocardiogram, as there are phases, it's the same heartbeat over and over again, you can definitely split one segment in one phase of the heartbeat. And actually, this is why ECG started to work way earlier than EEG. It's because ECG have been handcrafted, features have been extracted in a handcrafted way. to find the beginning of the heartbeat, the end of the heartbeat, the maximum and the minimum values, etc., and the times between both. So this is the answer for the ACG. It's possible because it's the way it's done usually. There are maybe other applications, but there are so many that I don't know all of them. But so the same questions of yours applies for EEG. Whether is it good to have data augmentation by splitting? This is what some people do. Yes, of course. The only problem is that when you do that, be aware not to do any data leakage. Because if the same person is in the train set and in the test set, there's a serious problem. And we've recently wrote a review of these progression models, and half of the papers are introducing some data leakage for such reasons. If you have launched data, some workers, ML practitioners, split the data into augment the data before doing the split. So at the end, you may have the same person within the train and testing set, and this is not a good idea at all. I mean, to have better results, but you're wrong. I have a question for anyone who knows. Is there any way to remove the questions once it's answered? Could you share materials for the suggestion Q&A application? Well, again, as for any applications, I've add only at the bottom of the slide. I'll share them. One paper, usually it's 2 or 3. Those papers have 2 objectives. The first one is because they are sometimes representative of the whole field and what's going on within it. And the second one is when you have one article, it's very easy to go to the other one. So I would say that materials goes at looking at the literature of the application I gave, but if I misunderstood your question, you can resend me a PM. What do you think about the idea of transhumanism and brain digitalization? Well, to be honest, I'm very, very, very involved in another topic personally, which is the relation between economy, energy, and climate change. And I have no reason to believe that it's going to happen for totally other reasons. Personal belief. And as for the brain digitalization, it actually already started. I mean, look at your hard drive. Do you remember any phone number of any of your colleagues? Or even your grandma or whatever. And there are a lot of tasks that are already handled by our computers. I mean, I'm fine with it. Does synthetic data work for AI medicine? Because I heard that synthetic data doesn't work for some type task. What do you mean by synthetic data? Synthetic data may have different meanings. So either it's a data augmentation strategies. It sometimes work, sometimes doesn't. The pools of data, okay, I've never encountered papers that properly show that their synthetic data are validated. Let me explain. When what's his name? Krasinensky, the guy that wrote the first data augmentation technique for neural networks. He said, okay, let's mirror them, let's translate them, let's doom them. Okay, let data augmentation. Okay, yeah. So he said, okay, a cat is a cat no matter what, how we see it, if we zoom it or not, which is okay. On the other hand, if you synthesize some data for brains or whatever, you have to prove, before to use them, you have to prove that the data you've synthesized are somehow correct. And to be honest, I haven't seen that happening. There have been, okay, this is not true, I'm thinking in the meantime, but there are some experiments with GAN data that are providing some synthetic data, but I haven't seen them used for now. Okay, and this is not even true. I've synthesized some data 2 years ago during my PhD for patients, as we had a generative model and it was working. So I'll answer this question again, coming back to the question about frequentist invasion approach. A lot of ML algorithms are black boxes. Even if you understand them, those are only rules that without any prior knowledge. It's not exactly a frequentist approach, but you don't have any stats in them. A neural net has no any distribution, no any nothing. It's just deterministic rules. And in that sense, it's very hard to do synthetic data as you don't apply anything which is close to a distribution or a random variable. On the other hand, less people are doing such thing, and we've been doing that, but other as well. The variational autoencoder has been exactly made for this reason, is to have not an autoencoder, but an encoder that has a generative part with a random variable. So from that perspective, but it's a very tiny community doing that. And it really, 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 really depends on the task. So I think that I've answered everything. If you have a question, you can still ask in the second lecture. And let's see you in 20 minutes or 30.
Все, Ігор, ми можемо починати.
Починати мені?
Так, я зараз напишу в Slack, що лекцію почали. Все, дякую.
Okay, so let's start the second session. Yeah. So just to sum up what we've just said, it was basically a brief overview of the different aspects in machine learning that are very specific to healthcare and medicine, in the sense that machine learning, that healthcare provides data that are quite complex, that are very diverse, and that need proper workarounds. And also the applications are very particular and very diverse as well. And so we've been through some of them. We've been also, I've given a short overview of the main difficult and I would say strategic challenges and issue that needs to be tackled by machine learning. And by the way, even though some of them has already been tackled by the machine learning community and the applied math community, on the other hand, some are still open questions and thus full of opportunity for those of you that like to tackle such challenges. And now, after this overview, what we'll look at is more precisely medical imaging, as it represents, I don't know, maybe 80%. It's a random number, but a very large number of all the applications of machine learning in the healthcare community. So as for the other type of data, I'll explain what medical imaging data are, what are the challenges that are specific to this type of data, and what are all the different applications that can be tackled thanks to that. After concluded that part, I'll try to make you understand of what I started to talk about during the Q&A questions of the previous lecture, is this change of paradigm that we have to face when working with medical data and within the medical environment. And I'll briefly conclude with future trends and a conclusion. So medical imaging data. First, I do not refer to data to images that were taken by a phone, but to medical devices used to get a specific data of the body or a patient. So there are two types of medical imaging data. On the one hand, you have structural imaging data. Those data inform about the structure of the brain, or it's a brain, but it can be anything, the structure of an arm, of a bone, of anything. And on the other hand, you have the functional imaging, which informs you about not the structure, but what are the changes that relates about the functions and the activity of the body or the brain or any part you'd like. And those are very, very different. It's very important to distinguish both. In structural imaging, you don't see what is happening. You just see the structure. On the other hand, with functional imaging, you see what is happening, but you don't know exactly where, right? And so at least it's important to keep in mind. So I'll start by explaining what the different medical imaging data there are, because there are many, and people often are confused about the differences between them. The first one, the well-known one, is a scanner, the X-ray. So nothing more than having X-ray going through your bones, your body, whatever, and having a receptor on the opposite side and look at whether the rays that have gone through your body or not. And based on that, you have an image. So this is the first medical imaging data that have been done by Röntgen. They kind of invented the procedure. And this is a scanner of his wife's hand. Yeah. And so now you have more detailed scanners here of the mouth and of the teeth. And so depending on the tissue, some tissues within the body makes the lights go through, some not, and this is why you have such an image. You can go to a bit more complex data. It's called computer tomography, CT scan, computer tomography scans, and it's basically the same principle, but where the emission part and the detector circles around. So you start emitting some X-ray from different parts of your circle and you intercept them on the other part. And once this is circling, you'll somehow know where the data you're looking at is within the 3D space. And that allows you to have similar images, but instead of having them in 2 dimensions, you have here now images in 3 dimensions. This is almost like the same principle. Another sort of structural imaging are structural MRI. Those are very different from the previous one. Here, instead of having light, like some light going through the body, here you have a magnetic field, which stands for M of MRI, magnetic resonance I. And this device changes the quantum property of the atoms that are within the body. And it records these changes. And depending on the changes, it can know whether which type of cells or body or atoms or molecules it is. And so thanks to that, you have such images. And again, those images of the brain, for instance, cannot be done with X-ray, because if you have X-ray through a skull, it won't go through the bone of the skull, so you won't see what's inside. Whereas here, as you excite all the molecules and all the atoms actually of the body, you can see everything from a structural point of view. And the I stands for imaging, magnetic resonance imaging. You can do quite the same, but a bit more complex by exciting the water molecules and to somehow, thanks to electromagnetic properties, getting which direction those molecules can go. And if there is a barrier around the molecules, then you know that there is some sort of body barrier, whether it's a bundle or a vessel, etc. Whereas on the other side, if the fluid can go almost anywhere, that means that there is no obstacle. And so thanks to that, so it's called diffusion MRI because it's based on the diffusion of the water, and you have such images. So here you have a very beautiful image of the bundles, of the fiber bundles, which are basically the gathering of the neurons within the brain. Again, this is the structure, a different type of structure, but still a structure. You don't have the activity. And finally, there's the functional imaging. So one of the most well-known functional imaging type of device is the PET scan. PET stands for positron emission tomography. And basically, you have a molecule that would like to somehow check within the body where it's consumed and where it goes. For instance, glucose. And you attach to this glucose molecule a tracer, a radioactive tracer. And they'll, as they are tied together, they'll go within the body. And at some point, the radioactive tracer will disintegrate and will emit two particles in the opposite directions that you'll detect. And based on where you detect them, you can know where it has been disintegrated, and thus where the glucose or any other molecules went in the body. So in the glucose, you are maybe interested in the fact that the glucose goes in the brain, and then you can have the activity of the brain, or no activity in some cases. But the output and the images really depends on the tracer, because there are a lot of different radioactive tracers for a lot of different molecules. And finally, the functional MRI is also some type of functional imaging, as it looks at the activity of the brain. And it's quite complex to grasp, but in a nutshell, it looks at the variation of the magnetic field that are produced by the consumption of the brain. And so there are chemical changes and chemical things that happen in the brain and that you can detect as they produce a magnetic field that you can compute and detect. And so if you ask someone to do something while looking at while being in the functional MRI, you can look at the spots in the brain that are activated during the particular task. So here again, you have the activation of the brain, and this is why it's functional imaging. Okay, so what about now we have the data and the diversity of data. All of this data face different challenges. They're pretty much the same for all of those data. The first one, and that we discussed in the first lecture, is that they're those data are very, very high dimensional. In the sense that any of the data is in 2D or in 3D, and one size is of 128 to 512 pixels wide. So if you have that to the power of 3, you have more than 10 million pixel or voxel. Also, as imaging data is more of a full pipeline, in the sense that you first record the signal, then you transform the signal you get in the device to a real image, and then you extract the feature of an image, etc., etc. There is a full acquisition, reconstruction, and preprocessing algorithms that are in place, and that all are to some extent not perfect and add some noise to the initial data. And so you have to know whether what you see is part of the noise or not. And finally, also, and this is related to the noise, there's a finite resolution in the image you have. And it's usually not for all of them. It really depends on the type of data. But let's say it's a magnitude of 1 millimeter. And you have to compare it where the diameter of a single neuron is 10 micrometer. So way smaller. So you cannot see neurons, except for some type of medical imaging data that are more research devices, which have more powerful electric field or magnetic field that are able to look at more precise objects. And this is to tackle also the fact I have not included in the lecture, but you have to think about medical imaging in two different manners and ask for two different types of application. One application would be medical imaging data for real life usage to make a decision, to have decision support system based on the data you have. The other type of applications you would have are not patient related, but are more research related. And in that manner, you try to extract information from the data itself, no matter who the patient is, whatever. You just need one device, maybe only at one place in the world, to better understand how the brain works, or the bone works, or the heart works, etc. And for that reason, when you are in the second, in the first application where you have real world data that you try to work on, you face the fact that the data are not processed in the same manner. The protocols are different, but also the machines are different. And the hyperparameters tuned by engineers are also different. And I'm going to tell you an anecdote I was told maybe 2 or 3 years ago at a conference. And it was actually very interesting, as one of the speakers said that they had a very, very good result on some prediction of heart attack, or I don't remember what the data was. And they went a bit further, saying, okay, now that we have such a good result, that was really a breakthrough, let's try to reproduce it and to validate it on other data. So they waited for another year to collect the same data on the same machines with almost the same patients, and they did the same protocol, everything's the same. And what happens is that the results went back to the random algorithm. So basically, a random chance on having good or false. And so they actually wonder where the difference from the initial data came from. And they started looking at the patients, and they were almost the same. They looked at the machines that were the same. The centers where the patients were included were the same, etc., etc. And the only difference they found so far, maybe the story is different because maybe they found a real difference, but at that time, the only difference they found is that Siemens, the constructor of the MRI machine, the MRI device, updated the software. Everything was different except for an update of the software that was reconstructing the data, the feature, the MRI from the signal. And that led to different results. And actually, for those of you that are interested by those things, there are some people have done some attacks, they're called attacks on neural networks, by trying to know what type of noise can you add to an image to change completely the prediction of a neural network. So if you're interested, drop me a message, I'll send you some links to that. And it basically, one of the well-known example of that is to add some pixels on stop signs on the street, so that your car, your self-driving car, doesn't see the sign and just go through the stop sign. Another interesting challenge is the fact that the patients are not aligned. What does it mean? Is that if you do an MRI or scan or any medical image of one person and then of another person, you can't say that 2 pixels on the same pixels, same XYJ of the 2 images represent the same part of the body. This doesn't seem as a problem now, but in the applications, I'll tell you why it's really a big burden. Yeah. So let's keep that part for now. What I also showed you is that with images, you cannot do some classic Euclidean operations, plus, minus, division, and multiplications, because images does not directly belong to Euclidean space, but subspace, a submanifold, on which some of the super operations are listed, but not all of them. And you first have to find the subspace where the data live on. And another interesting issue when you deal with images is how would you compare them? Let's say you have an image of a cat and an image of a cat that is translated by 3 pixels to the left, or let's say 10 or even 20 pixels. To you as a viewer, you'd like to say that the images are exactly the same. The cat is here, he's exactly the same, so the loss is almost null. However, if you use generic losses such as L1 or L2 or any losses that you may know, you'll have a very, very high error, which is something you don't want. And you have the same representation for forms or meshes, where once you compare, what is the mathematical operation you'll use to compare those two forms? And this is the loss. And actually, there is even a community that works quasi exclusively on losses to try to understand how to compare images or features extracted from images. Okay, so now from all of that, let's go now to what's probably an interesting part, the application of thanks to medical imaging in healthcare. But just before that, for those of you that are not well aware of deep learning, I'll add just a few words so that we're all on the same page when we speak about deep learning and different architectures. Standard machine learning algorithms are composed of usually two separate parts when they deal with images. The first part is that you have your image, you extract the feature, such as the number of blue pixel, the number of gray pixel, the histogram, the gradient, the fact that, I don't know, whatever you can extract, which pretty much corresponds to the Photoshop extract features you can have when you change some sliders. And once you have these features extracted from the image, you have an algorithm. Here's an SVM, it can be a random forest, or linear regression, logistic, whatever. And those two parts make your pipeline. You extract your feature from the image, and then you have the algorithm. On the other hand, what is very interesting when you use deep learning is that it integrates these two parts simultaneously. So on the one hand, it has data descriptors. It has some mathematical operations to describe the data. And those data descriptors are included within the algorithms. What you have to remember is somehow, if you compare, this is a well-known picture that tries to compare both. In one case, machine learning first extract the features and then do the classification or any sort of algorithm. Whereas in deep learning, the feature extraction part is included within the algorithm, which is very important, as if you do the feature extraction separately, you don't know if it's suited for your problem at hand. Whereas in deep learning, you know that both are done together. So the features that are extracted are those that are the best to do the specific task you're working on. The second thing I'd like to add on deep learning is that when we refer to CNN, it's convolutional neural network. So those are neural network that are used that uses raw images as input. So you can take your medical image as input. You do not extract any features from it. And the last architecture that I'll briefly speak about is the autoencoder. It's a weird architecture where you input an image, but you can actually input whatever you want. And what you'll output is exactly your input, or at least you'll try to do so. And what's the purpose of that is that when you represent your image with 28 times 28 pixels, you'll reduce the dimension that represents your data to a smaller dimension. That's the bottleneck in the middle, which here is two-dimensional, which means that any of your image can be represented by only two values that you can then decode to have your reconstructed image. So once again, what you have to remember is that you have the autoencoder is a neural network that reconstructs an input as close as possible from the input. There's an output, sorry. Okay, now that we know that, last year, Daniel Rickert, that is a well-known scientist in the field of medical imaging, had a very interesting way to represent what are the different types of medical imaging applications, and they are ordered by their level of diagnosis support. So the first one do not help in the diagnosis support, whereas the last one are very towards heading practitioners. So what we have first are image acquisition and reconstruction. So when you acquire a signal, you try to reconstruct it. Then, once you've reconstructed your medical image, you try to enhance it by denoising, by having a better resolution, etc. From that, you have an interpretation, which is a representation more meaningful than just pixels. Maybe you have clusters, maybe you have some areas, you can segment some part of the image. Then you can quantify some of the features that you've seen on this image and use them as a biomarker for other machine learning tools. Those biomarkers can be used to detect or diagnose a disease. And finally, which is the holy grail, is to have a disease prediction system that based on the image is able to predict what's going to happen next. And this is exactly the path I'll follow to show some applications of medical imaging data and neural net architectures. So first, image reconstruction. Before, when we talk about the PET data, remember, we were acquiring on the receptor two particles on the opposite direction, and from that, we knew where the particle was disintegrated, and we were able to reconstruct the image. So there was some sort of mechanistic model that we're saying, based on what I record through time, what is my initial image based on the input image. So this is the recording of the disintegration through time. The y-axis is actually time. So at each second, where do you, on the circle, where do you have some disintegration? And what you've done is you reconstruct your image thanks to a mechanistic model. And so what people have started to do, thanks to neural net, is basically do this reconstruction without any model. So go from the input image to the reconstructed image directly thanks to a convolutional encoder-decoder network, some sort of autoencoder, a bit different as it's not the same image at the end. And the point here was to say, okay, we don't even need the physical model to say how to go from the input to the reconstructed image, which is actually not perfectly true. Why? Because when you train the model on the pairs of data, the reconstructed image was reconstructed thanks to this physical model. So when you train this convolutional network to redo this operation from the input image to the reconstructed, you actually learn the model itself. Then there was some question about synthetic data. So it's used, and basically some people are trying to have a first initial image and to synthesize a new modality based on the first image. So here you have MRI and you want to synthesize CT scan. So on the left panel, those are MRI, on the right, the CT scan. And you see actually that on the CT scan, you see only the exterior part of the brain. And so those type of translation going from the the MRI to the CT scan is done. And so the middle column is the synthetic CT scan, whereas the right column is the ground truth you want to reproduce. So the architecture here, which is a UNet that we'll talk about a bit later, is able, given one modality, one medical image, to reproduce another medical image. And why are people trying to do that? First, it may be less expensive to record some of the images compared to the other, but also you don't have all the engineers able to control all the machines everywhere, so it can be less costly to just synthesize the given image based on another one that is less costly, easier to get, or that maybe just is the only one that someone can interpret in the hospital or in the environment you are. Then, this is the second part. Once you have your image that you've reconstructed from your input signal, you may want to denoise it. So this is an example of how to denoise an image. The middle image is a very noisy image that, thanks to a GAN, a generative adversarial network, is able to reconstruct the initial image without the noise. I'll skip what a GAN is, but for those of you that are interested, the reference is at the bottom. Just remember that those neural networks are networks that try to fool another network by producing images that are close to the reality. So that's two networks fighting, one trying to say if the image he gets is positive or negative, is true or false, and the other one is trying to fool the first by simulating images that are real. And so this generator can take input data to make them even more realistic, and this is what he produced, and this is how he denoises the MRI. Another type of image enhancement is called the super resolution. The super resolution is a fancy word to say that we'll go beyond the resolution of the image we got. As any device or microscopy has limitation due to optic reasons, but also to image treatment or algorithm reconstruction, we can improve this resolution thanks to what's called the super resolution. And this is done, for instance, here, it's a fluorescence microscopy, where the resolution of the input here is enhanced to have a better resolution as this one, and this is the ground truth. And actually, those type of super resolution algorithms are starting to be in your smartphones. They enhance the zoom or just improves the pictures that you're taking on your phone. So you can do the same. And why doing that is basically to, if the device you bought to have some medical imaging has been bought 10 years ago or 5 years ago, is not good enough compared to other devices, you can add a new layer of super resolution to improve the resolution of the image you have. Another enhancement is the registration. So you probably remember that I told you that one of the challenges of medical imaging is to be able to register two images one to the other. Is basically be able to move one image at one position to another position so that you can compare two images. Why would you do so? So why would you so? One of the reasons that you may think of is to help deep learning algorithm to converge. If all the images are aligned within the feature space, they all represent the same thing, maybe it's going to be easier for the network to converge and to find meaningful results. But this is actually not the real reason, and actually not the first one. The first one is to be able to compare patients. If you want to compare patients, you have to know that the pixels can be compared. If there is a hole here and a bone somewhere else, maybe there's a lesion. And to know that there's such a lesion, you have to, again, compare those images, and you can do that only if you've registered the image before. The last reason is to fuse data from different medical imaging. Remember, I told you that there's, on one hand, structural imaging, on the other hand, functional imaging. The structure doesn't tell you anything about what happens within the image, whereas on the other hand, the functional tells you what happens, but doesn't tell you where. Maybe it's in the liver, but maybe it's just around. Just imagine you have a tumor. You have to know which organ it is if you want to do an operation. And such thing is not possible just with a functional image only. And that's why registration is so important. You can have two different types of images and you register them onto the same template so that you can have a layout of one image over the other to understand where the function is happening in the structure of the body. And so you probably see that here you have the original functional image that you register so that you move slightly and you fuse those two images to have the last one. So how do you do image registration? There are actually a lot of different possibilities that have been worked by a lot of scientists in the past, maybe 15 years. Here is you have your first image. It's called the identity, and you don't do anything to the image. One mathematical operation you can apply to this first image is it's called a rigid transformation. It's basically a translation plus a rotation. Then what you can add as a mathematical operation on top of that is a similarity operation, which is basically a scale, zooming in or zooming out. And finally, you can use an affine function to deform a little bit your input data. And finally, you can do real deformation that are way more complex. From that, we can look at the degrees of freedom of all of these mathematical operations. The first one, the identity, well, has no any degree of freedom. The rigid has 3 to 6, depending on what you account for. If you have a 3D image, you can have 3 rotations and 3 translations. Then if you have a similarity, you can zoom in, zoom out in the 2 or 3 dimensions. Only in one direction. Then the affine transformation gives you a 6 to 12 degrees of freedom. And all of those 3 first operations, rigid, similar, and affine transformation, are all linear. And it comes to the part that then you may have to use nonlinear transformation, such as the deformations. And in that case, you have very, very complex model that may include thousands to millions of degrees of freedom, which is okay if it's interesting for your application. But on the other hand, you don't know much about the identifiability of your model when you're using such a high number of parameters. To say differently, to go from one image to the second one to register, there might be one way or 2 ways or 3 ways, and you don't know which one's the best. For those of you that are interested again in this type of applications, there is a well-known algorithm called voxel morph that was presented 2 years ago at CDPR, and which is one of the state-of-the-art algorithms for those type of registration with deformation. And again, a last thing I'll add is sometimes when you register, you are maybe not interested in the registration itself, but in how the input image was deformed to go to the template. So let's say that you have 2 images that you'll register onto the same template, then you may be interested into the amount of deformation that was used for the two different images to register on the same one. Now, let's go to the next part where once you have your data, your signal that is well reconstructed, denoised, maybe with a super resolution, you try to get meaningful information from it. You have just pixels, and now you want to know what's on the picture. So that's what segmentation is. You segment your image to first know where things are, where cells are, for instance, or where mitosis is, or where mitochondria is. It's just where. It can be yes, no, where cells are, where they aren't. And after that, the next step is to know what is on the image. If you have different cells, you maybe want to know what, once you know where the cells are, you want to know for which class it belongs to. And for that, there's one like king of neural networks, is the UNet, which has been introduced 5 years ago, but has already more than 12,000 citations, and that lead to a lot of ad hoc specific UNet architectures for specific segmentation or construction. But the base of all this UNet, of all those neural networks, is the UNet, and this is the architecture. I'm not going to go into the details, but the idea is that you really compress the information before decompressing it. And on top of that, you apply some copy of your input images or after some layers directly to the output. So it's a UNet because you compress the information like that, and you decompress it, and you copy some images here directly to the output here. And once it was released, even at the beginning, the first architecture it was actually winning a lot of different competitions. Next, let's talk about how to quantify once you know how to segment, or even if you want to know what's on your image, how can you do that and quantify the biomarkers such as volumes, such as thicknesses that are on your images. So this figure is the figure of an autoencoder. Again, you have an input, you compress, decompress it, and the goal is to reconstruct an output which is as close as possible to the input. And in that case, usually people do that to denoise the input data. When you compress your information, you can no longer store the noise. So when you decode without the noise. But what if you look at the noise? Basically, maybe the noise has a structure. Maybe the noise is an information, an artifact that you want to know about. So here is your input image. Here is the ground truth segmentation that we will compare to. And this is actually the noise that has not been reconstructed by our autoencoder. So when you do the input minus the reconstruction, this is noise, and this is very close to the ground truth segmentation of some tumors that they are on the image. And this is a way to segment your data as well. Now, going a step further, let's move on to the detection and diagnosis of disease. There are many examples out there of researchers doing that, startups doing that, industries doing that. I'll just highlight some of them that are well known by the community, as they were the first to prove that deep learning was able to do something in medical imaging. The first one is the breast cancer problem. There's a public data set that was tackled by a lot of laboratories around the world. It led to different, way more paper than what I wrote at the bottom. And the goal is based on a mammography to get if there is a tumor or not, And if yes, where it is. And maybe if there are multiple malignant nodules, where they are. The second one is the diabetic retinopathy, and you may have heard about it as it was a Kaggle competition won by Google, I guess, at the time. And the goal was to, so those are pictures of the bottom of the eye, and based on a given type of diabetes, there's a change of the bottom of the eye that you can detect to know whether the person has diabetes or not. And so this is one of the architecture that the winner used to win the challenge. But what was very interesting is when Google released their paper about the fact that they win and the model they've used, they also said a very interesting thing. Basically, we don't have enough data, so they paid a lot of practitioners, ophthalmologists, to label new data, like 1,000 or 10,000. I don't remember the amount, but they were not particularly thanks to their architecture, but because there are way more data to feed their neural network with. And for that reason, they were able to have a more complex network to look at more particular features on the image itself. And here, I think it's one of the architecture that is used for other topics in object detection. It's called the Inception V3 model, I think. But I'd like to raise a point before going any further. I've been talking a lot about deep learning, but some people thought, okay, now that we know that we can do something, thanks to that, we can detect, diagnose, and enhance, would like to better understand what's going on and basically interpret the data and know why a given architecture is better than another. And so what some people did is to have different architectures with very different approaches. So the second one is the SkipNet architecture, and they've compared it on some data sets. And what they see at the end is that on the task of comparing Alzheimer brain MRI versus cognitively normal, both algorithms were doing almost the same. And so other teams looked at the same, but they do not only compare deep learning architectures, but they also added comparison to more standard techniques. And what they saw is that here the SVM was better than the other CNN network, even though with the standard deviation of the balanced accuracy, all the models are quite similar, except for the 2D slice level CNN. What I'm trying to say here is that deep learning is not a holy grail for everything. During the Q&A, I said that start with deep learning as it's usually easier at the beginning, but where you're in a research program, when you have time, when you're under a PhD or even under R&D, do not forget to at least try other algorithms to know what they're giving, because they can be as good as, and also sometimes when they are as good as deep learning, they are not as good as on the same data. So they can be interesting to combine different approaches. And in the same sense, the lack of interpretability, some labs and some teams have looked at, can we say why a neural network is saying what it's saying, which part of the image it's looking at to do the prediction. So this is an example with cars to know which car it is, and you know which part of the image it's looking at. And those maps are called attention maps or saliency maps. And it has been done either, so here on the brain to look at which part of the image the neural network is emphasizing on to do a prediction. But there's also another way to do that. Basically, you mask your input image, you erase some of the input pixels or voxels, and you look at what are the pixels or voxels that decrease your prediction the most. And in that case, those would be the pixels that are the most important for your prediction. It is a bit more precise than just looking at what's the most important pixels, as in the first example, for the reason that one pixel after layer gets through a lot of different neurons, and it's hard to keep track of what it's actually doing within each neuron and each linear combination or feature map in the case of a CNN. Yeah. And finally, and this is actually in the spirit of what I showed you about disease progression models, people are trying to simulate the longitudinal progression of the disease saying through this manifold. So the manifold is the subspace of possible observation. As I told you, you cannot see all the images of your baby. There are only a subset of images that you can see. And this is to some extent called the manifold. And so people have done that to predict the progression of MRI data. Instead of just predicting the label, they are predicting not only the label, but actually the whole feature itself. And it can be more interesting for a practitioner because on the one hand, let's imagine you're a neurologist and you know that the person in front of you will have Alzheimer's disease next year. Or on the other hand, you'll know that its cognitive score will be very high or very low. You have more information in the data rather than one label. Because in the data, you can look at different parts of the data and better understand because it provides more information. And so this is one of the examples of having 3 images and trying to predict and to simulate what the future may look like. Okay, so that was it for the medical imaging part. Now let's go to something that I believe is really important once collaborating with medical practitioners, and when also they collaborate with us. Why I'm speaking about that is before medicine used only, or not every time, but was able to use only was example to find a rule. For instance, in the past, almost a century ago, some people have lost part of their brain. And when they've lost their part, this particular part, they've lost the speech or the vision, etc. And from that only example, doctors were able to know that this part is related to this particular task. They were needed only one example. Whereas in ML, machine learning, we actually use a lot of examples to draw conclusions, which is very different. So let's imagine the doctor that instead of looking at one MRI actually sees that. How this person would draw some conclusion the way machine learning does. And so basically, there's a need of changing the way doctors look at this data and understand them, but also for us to provide information to practitioners, as they are not mathematicians, not computer scientists. But can we do that? Can we meet the best of both worlds? So basically, first we have to, to me, it's the next step that we have to achieve, the next challenge if you want to go further in the collaboration between 2 worlds. But we can also do that not because we have to, but because we have already done it. And to highlight it, I'll give you an example of, this is a very weird example, but you'll understand a bit later on. The tide of the water. So basically the water going up and down under astronomical condition is a very well-known process. But actually what you may not know is that there are no physical models that are able to accurately predict the effect or the magnitude of the tide just based on the physical condition. You probably know that from one city on the coast to another, the magnitude of the tide may be very different. And this difference is not known from physical models. So I'll let you think about it for 10 seconds as to why and how do we know the amount of tide, its magnitude from one city to the other, if we're not able to do it from a physical perspective. Well, so basically, the reason is not that we know how to model it, but we have seen a repeated number of times. And so as we have done it a lot of times, we're able from a statistical perspective to know the magnitude as we've seen in the past. And there is a repetition in that helps us understand what's going on. So I'd like you to keep this example in mind for the next few slides. I'll try to sketch what a model is from a biological perspective and what it is from a statistical perspective, and why those two are different, and why the medical practitioners and the ML practitioners have to understand that to better speak and collaborate together. So a phenomenon, let's say that you have a given phenomenon and you have some observation of this phenomenon, correct? So what we do is, based on this observation, we try to understand the underlying mechanisms of the phenomenon we are observing, whether they are physical, whether they're chemical, whether they're social or economical. This is basically what a model is. So if you look at day and night, this is your phenomenon. Your observation are the earth size, the sun size, the distance between both. And you'll model your phenomenon thanks to a modeling field, which in this case is astronomy. We don't use, not everyone uses the same tools to do the model. So in some cases here, it's the astronomy. If you have money transfer between one person to the other, the observation you might have are the amount of capital, the amount of work, the amount of innovation you have between countries. And the modeling field to understand how the money is transferred from one state, for instance, to the other is the macroeconomy. Heart beating, more of a biological model. To better understand the phenomenon, what you'll observe are the ventricle size, the blood flow, the heartbeat itself, and you'll have a mechanistic writing of your phenomenon to understand why it's getting faster or slower, etc. So this is what we'd like in a biological setting. And as there's a link between the phenomenon and the observation, we better understand the underlying mechanisms thanks to causality, but also correlation, what the consequences are, some sort of a links. And as soon as there's one observation that cannot fit the model we've written, we find another one. And this is basically the reason why Galileo died. There were observations. So before him, people were saying that Earth was in the middle of the solar system. And they made very, very complex models of our solar system to make all the observations fit those models. But it was getting absurd to a given point that Galileo said, no, It's their way. It's an easier model to have the sun in the middle, the Earth around, all the observation fit. Well, and he had some problem afterwards, but this is another story. But sometimes it's not that easy to go from the observation to the phenomenon, because there are no modeling field that allows you to go from the observation to the phenomenon. And such modeling field does not exist, in particular where you have micro and macro elements that you want to combine together. Let's take some examples. If we want a model that explains both the infinitely large and infinitely small physics, such as astronomy and quantum physics, there are no yet modeling field to bridge those two observations. Same goes for macro and microeconomy. Yet those are two different fields because they explain each within its scope better what they are trying to model. The same goes for the brain. There are a lot of teams, biological teams working at a molecular level, at a cellular level, at the genetic level, but the mechanisms are poorly understood and the relation one with each other are also poorly understood. But moreover, we don't know how all those molecular or cellular phenomena output what we see as phenotypic observation, and what people do, what people believe, what people think, how they talk, etc. So what we have is only one way, from the phenomenon to the observation. But does it mean we shouldn't do anything? We shouldn't model at all? No, this is not true, because what we can do is to find a model that is not related to the phenomenon that is producing this observation, but a model that describes the observation altogether as a whole. It's basically relations between all the observations that fit together. And in fact, this is what we do all the time. We have internal models that makes the observation we have of the world coherent. Let's imagine you are in a plane and everything's fine. All the observations are okay, there's some noise, there is a baby screaming, you got your Coke or your coffee, etc. That is fine. All the observations match all the others within your internal model. But as soon as one observation does not belong to your model, you know there's a problem within your own model. And such observation can be a fire in the reactors. So this observation will not fit the other one anymore. And you know that something is not good. And your model doesn't work anymore and you start screaming and, well, might be a problem. But this is it. This is how we work. We don't know why the fire started. It goes the same for a lot of things. We don't understand all the phenomena that are producing the observation. Nonetheless, we have this observation all coherent. And this is basically what a statistical model is. And this is what machine learning does. It's just trying to make the observation coherent one with the other. So when people try to work in healthcare, machine learning scientists, I mean, they're just trying to model it in a way that the observations are coherent one with each other. It's not possible to have, for instance, a brain bigger than the head size. This is a weird example, but it's just to illustrate what I'm saying. And so in this statistical models, we have to do some hypotheses about the phenomenon that we don't observe, but still, we make some of them at least to have a prior description of our model. This is also a try and error modeling process, as we try to have a model that makes all the observation coherent. And once we have one observation that is not coherent with the other one anymore, we rebuild or rewrite a statistical model, etc., etc. Then it does not mean that the statistical model, and it's important to keep that in mind, it does not mean that the statistical model is perfect nor true. it just means that it have the data that we have so far coherent one with each other. Galilei, for instance, has not the perfect model. As soon as Einstein discovered relativity theory, not Galileo's, but Newton's model and equations were not good anymore. I mean, not in a larger scope. And finally, it's important to remember that from basic statistical models, even though some people are trying to go beyond this thing, but we can only see at the first glance correlation. No causality nor causation between the phenomenon and the observation. Even though there are a lot of things going on to try to understand what is the link between the observation and what's the causality. Okay, so that was it. It was just to conclude about the fact that we have to be very careful about what we call a model and what biologists and doctors want as a model, meaning they want the link between the phenomenon and the observation, whereas what we can provide, as for now, is more of a statistical model, a relation between observations. As for the future research that to me are important in the future, and some teams have grasped those ideas, is basically first have new algorithms, methodology, applications, that's for sure. But that has always been like that. There is, to me, something that is a bit more important than that if you want to transfer from research to clinical data or real-life applications. And those, to do that, there are 3 steps. The first one is to validate what you're doing. Validate means to be sure that you're doing what you want to do, that you don't have data leakage, that you've done a good cross-validation, that everything you say has been assessed or proven within one cohort, with one data. The second step is to replicate it to new cohorts. This is very important, as in these cases, this is where you might see that there are differences between real data and clinical data, between different centers, between different medical imaging devices. This is important. And the last one is the interpretability. No one, at least not a lot of persons, wants something that tells you a number without being able to interpret it. And this is somehow the way that attention map took to better understand deep learning algorithms. And to illustrate this thought, I'll ask you to, how would you think an algorithm would separate sheep from penguins thanks to these pictures? Well, for a human, it's actually very easy. We know what a sheep is, we know what a penguin is. A penguin is in 2 feet. It has a yellow color. It's all black on the top. This is fine, but this is not what an algorithm will see. And to illustrate also again that, I'll show you some data where the algorithm would fail. What the algorithm will look at is basically the background. If the background is green, then you have a sheep. If your background is white, then it's a penguin. And once you have data that you haven't seen in the past, you won't be able to generalize what you were saying. And this is the reason interpretability is so important. It's not only to interpret, but it's also to be sure that you understand what the algorithm is doing and that it's not doing something stupid. So this was kind of a conclusion. So I hope you enjoyed the lectures. I hope you now know what the particularities of these fields are. There are a lot of opportunities. It's a very exciting field for a lot of different reasons and for a lot of different perspectives. I hope that you are now aware of that, and thank you. And if you have any questions, I'll be happy to answer them. Well, I'll actually let you 2 minutes to think. There is one question, maybe 1 or 2 more. I'll just share in the previous questions in the first lecture, there was a lot of questions about what's going to happen next in the future. What are the trends? If it's possible to have a virtual brain, neural links, etc., etc. I'll just read you one title of the New York Times, and I'll let you 10 seconds to wonder when it was written. So the text is, the Navy revealed the embryo of an electronic computer today that it expects will be able to walk, to talk, to see, to write, to reproduce itself, and to be conscious of its existence. The Navy revealed the embryo of an electronic computer today that it expects will be able to walk, talk, see, write, reproduce itself, and be conscious of its existence. And so this was written. It can be the title of any media as of today, but it was written about perceptron, so the ancestor of neural network, in 1958. So, well, if you think that there's a 80 years old delay between what's written in the journal and what's happening, I'll say that Neuralinks won't be able to do anything during my lifetime. And the only question that was written is, can we in this way synthesize ECG or EEG? And what about text data? Is it possible to generate realistic? Well, I don't know what in this way is, so I don't remember what I said almost an hour ago. If you can precise your question, I'll be happy to answer, but I'm kind of lost. What do you think about the project of MIT about cough analysis to detect COVID-19? Well, disclaimer is, I decided not to read anything that is related to COVID-19 and research. It's kind of a joke, but there has been so many research written in the past 6 months and so many papers and that it's hard to know what's true or not. So I decided to take a step back, and I haven't checked what they'll do. Oh, okay. Can we, in this way, with the encoder and decoder, synthesize ECG or EEG? And what about text data? Okay. Okay, so the fact is that with the encoder, decoder, which is not exactly, it's not, So when I was speaking about synthesizing CT scans from image, it was done thanks to the UNet, which is whose intention is actually to keep the same structure, which is not the case for ECG or EEG. I mean, I don't see how you can produce an EEG from an EEG that would be different. Those modalities are too different. I mean, it seems to be the same for text data. I mean, I feel that there are ways to synthesize ECG or EEG or text data, but not in the same spirit as it is done with the medical imaging applications when you go from one modality to the other and they're close one to the other. Here it's very different. You try to go from something to something totally different. For EEG and ECG, actually, I don't know how people synthesize data. What people do with medical data, if you know that there's a link between, you can feed an autoencoder with medical data, for instance. This is actually one of the theoretical way to synthesize data. Actually, almost any of them is to have a variational autoencoder. If you have a variational autoencoder, it's supposed to learn the random variable that can generate your data. This is in theory. In practice, what happens is that your latent space is too empty, even though it has random variables, it's too empty to learn anything at all. So it's not possible to use it this way, except for very particular tasks. And what's your prediction on climate change rates? We'll be able to make it for another 50 years, given current state of things? Well, no. I mean, at least in the literature, in the scientific literature, nothing tells you that we can. And by the way, there are birds that have been seen flying at almost more than 10 kilometers. Well, I think I'll end this session. So thank you very much for your attention. If there's still people in the room, it's kind of sad that we haven't met in person. If you have any questions now or anytime, do not hesitate to reach out, LinkedIn or PM or my website, whatever. I'll be up to answer. Yeah, and it was very fun having you. See you.
Thank you, Igor.